This is the University of Hawaii Maui College, the college on Maui.
Aloha and welcome back to General Pharmacology. We'll just move right to the PowerPoint slides and roll right along with the information that's on Exam 4. I think I brought the PowerPoint slides with me. Oh, there they are. Today we're going to talk about the immune system and the study of the immune system, immunology, the third lecture on the fourth exam. So let's talk about immunity. Immunity is the body's ability to recognize infectious disease-causing organisms like bacteria and viruses and then protect the body from these organisms. We'll have all sorts of interesting definitions of immunity in the forum section. If you just Google the words immunity, you'll get all sorts of interesting definitions. But I want you to think in this terms of immunity being the body's ability to recognize and then respond to them even better when it comes in contact with infectious organisms. So to study immunology, we're going to have to talk a little bit about hematology. Hematology is the study of blood. We're going to talk about immunology, the study of the immune system, the system that fights infectious diseases. We're going to talk about microbiology a little bit, the study of microscopic organisms. Hopefully you learn a lot of information in microbiology because I don't talk about that too much at all. Something I do want to draw your attention to is this word right here, epidemiology. This is the study of causes, distribution, and control of diseases in populations. Epidemiology involves studying populations and their diseases. Lots of statistics involved, but epidemiology is a very important concept in combating the plagues that have plagued humanity for a long time. I wanted to introduce you to this right here, smallpox. Smallpox used to be one of the worldwide scourges and killed many people. And then one day they came up with an immunization for smallpox, and we don't hear about smallpox anymore unless people are talking about terrorists and things like that. There is no more smallpox on the planet. Well, before 1979, smallpox killed 80% of the children who contracted the virus, and about half the adults. Since 1900, it was estimated that smallpox was responsible for 500 million deaths. In 1967, the World Health Organization estimated that 15 million people contracted the disease, and 2 million people died of smallpox in 1967. But after successful vaccination campaigns, the eradication of smallpox was declared in December of 1979. Smallpox did horrible things to the Hawaiian population as well as the American Indian population on the mainland. And so it was the development of immunizations that led us to be able to eradicate smallpox from the planet. So in the forum section, you're going to talk about people who helped develop the immunizations, the people who discovered the immunizations for smallpox, and I want you guys to take a really close look at that. Next time when we come back, we're going to talk about HIV and AIDS. We're not going to talk about that today, but there is a hunt on for the vaccine for HIV, the virus that causes the disease AIDS. And many millions and millions of people are going to die of AIDS before then. And my dream is one day I'll still be teaching this class 20 years from now, and I'll get an email from a student saying, why do I have to immunize my child against HIV? Nobody's even heard of that disease anymore. Nobody has it. And why should I get immunizations? Because they don't really work anyway. and I will have them go back and take a look at the history of smallpox to understand the history of immunizations. And I pray 20 years from now that everybody will be immunized against HIV, and that'll be another disease that nobody's really heard of. So let's talk about how immunizations work. Remember, we talked about locks and keys. We talked about drugs and receptors. The concept is very similar. And here's going to be the star of our show, at least right now. It's an antibody, this big Y-shaped thing. When you read about Y-shaped things in immunology, those are antibodies. And these are made by lymphocytes. And this end is the receptor. The top of the Y is the receptor. It's going to bind something to something called antigen, parts of viruses or bacteria or anything that can stimulate the immune system. And then this part right here is going to stick to either one of the cells, one of the white cells, parts of the body. This is going to attach to the immune system, and this is going to attach to parts of the organism that antibody is attacking. So the antibody is like a receptor. Here I have my locks, and here is an antigen, it's like the key that's going to unlock this antibody. An antigen is any part of any kind of, it can be part of the bacteria, part of a virus, anything that stimulates the immune system. So please do not confuse these terms and these abbreviations right here. Antigen is abbreviated AG, and it is usually a protein, any protein or any type of structure that can stimulate an immune reaction, a reaction from our immune system. The antibody, abbreviated AB, is that Y-shaped protein that I showed you, and antibodies bind to antigen to mark them for destruction either by phagocytes or stimulate the immune system. So know the difference between antigen and antibody, please. All right, so now that our antibody has bound to the antigen, here's our antibody, and the antigen has stimulated it, and then it can cause a reaction in the immune system. So activating this antibody it stimulates the immune system. All right, so there's a little introduction to antibodies for you. So antibodies could be free in the blood, and these antigen-antibody complexes can signal digestion by a macrophage. That's this right here. It's a big white cell. We'll do the rerun. Here is my antibody, and it is attached to an antigen, forming an antigen-antibody complex, and that can signal something like this, a macrophage, to come and consume the antigen. And we're going to talk more about that. Antibodies can be attached to lymphocytes, and this antibody-antigen complex can signal the lymphocyte to even make more of the same antibody. So if this antigen belonged to a bacteria or a virus, and this B lymphocyte recognized that antigen on the bacteria or virus, the antibody, or the B lymphocyte, can make even more antibodies to recognize that specific virus or that specific bacteria. So what we do with immunization is try to fool the body into making all these antibodies against certain types of antigens. So when you get a flu shot, and we'll talk about the flu in later lectures, when you get a flu shot, what they do is they take antigens from the flu virus and they put them in a vaccination, and then when your body comes in contact with those antigens, it makes lots and lots and lots of these antibodies that are going to be specific for those viral antigens, those flu virus antigens. And then the body now has this huge increase in the number of receptors, this number of antibodies that can activate the immune system in case of exposure to specific bacteria or diseases. And so this is basically how immunizations work. And so the next time the body is exposed to a specific bacteria or virus, that bacteria or virus is going to have lots of antibodies waiting to attack it, so they cannot come in and infect the body. The bacteria or the viruses are attacked by these antibodies, so that bacteria and virus can't attack us. So immunity prevents infection by increasing the ability for antibodies to signal the destruction of an infectious organism. If this bacteria came to infect us, it would be able to reproduce freely and cause all sorts of problems. However, when we have lots of antibodies that recognize that bacteria, it can signal the immune system, a white blood cell like this macrophage, to come consume it. And this is what we're going to talk about today. Before we talk about any of that, we need to study a little bit about blood, hematology. Now, I don't expect you to memorize all the terms we're going to go through, but one of the problems in pharmacology is they throw all these hematology terms around like you know all about them. And so I just wanted you to have at least some familiarity with the terminology as we go through it. So when we study blood, let's think about what blood does. It transports oxygen and carbon dioxide. We knew that. Blood moves nutrients into and wastes away from the cells. Blood helps clot the vascular system, plugging up holes. That's all the things that blood can do. But something else the blood does is fight infection, and that's what we're going to study today. And so what is blood made of? Well, the serum is the liquid base of the blood. It's made up of mostly salt water, very similar to the ocean. We bring the ocean with us as vertebrate mammals inside of our vascular system in the form of serum. Serum contains small proteins called antibodies. I think I showed you what antibodies were. They're made by lymphocytes, part of the immune system. And then something called complement. We won't talk about complement too much today. But complement's made by the liver, and complement is also proteins that are designed to attach to viruses and bacteria and mark them for phagocytosis as well. So antibodies and complement attach to foreign substances such as bacteria or viruses to help the immune system fight with these substances. And antibodies, like I said, are made by lymphocytes, a type of white blood cell. You might see white blood cell abbreviated WBC. And complements are proteins made by the liver. I'll tell you right now that antibodies are very specific for certain antigens. Complement is not specific. When antibodies are stimulated, it can stimulate lymphocytes to make more antibodies that are very specific for that antigen. But the liver just makes complement to go stick to any bacteria it can find. And so I wanted you to know the difference between antibodies made by lymphocytes and complement is made by the liver. Well, plasma is the same thing as serum, except there's some large clotting factors in plasma. I don't expect you to know too much difference between plasma and serum, but serum is the liquid saltwater base of blood with some small proteins. Plasma is the liquid base of blood with some clotting factors. Serum plus clotting factors gives us plasma. And then there are the blood cells. And the three cells that you should know about that make up blood are the red blood cells, the erythrocytes, the white blood cells, which are the leukocytes. Leuko means white in the medical business. Cytes just means cells. So white blood cells are leukocytes, and this is what we talk about when we talk about the immune system. We talk about white blood cells. And then there's platelets, the thrombocytes, and platelets are involved in clotting blood. So today we're going to talk a lot about white blood cells. Red blood cells, white blood cells, and the platelets, they're all made by the bone marrow. And then during fetal development, the liver also makes these cells as well. But in normal adult, red blood cells, white blood cells, and platelets are made by the bone marrow. The inside of long bones, long flat bones, is where the red cells, the white cells, and the platelets are made. Well, here is something called a peripheral smear, and I'll put those back. If we look at this peripheral smear, we see these red cells. Those are called red blood cells. And then these two purple-looking cells, we'll call them white blood cells. And then these little spots inside the black rings, those are platelets. And so when you look at a peripheral smear, if you take a little drop of blood and put it on a microscope slide and slide it out real thin, you'll have what's called a peripheral smear. And so this microscope slide is called a peripheral smear. And these two things right there, I'll tell you right now, they are neutrophils, a type of white blood cell, a type of granulocyte. And so see if you can see the red blood cells. They should be obvious. Those two white blood cells called neutrophils. And see if you can see those little purple spots, and those are the platelets. So let's talk about the neutrophils. One type of white blood cell is neutrophils. Neutrophils have many names. That's one of the problems with neutrophils. The medical business has given them many different names and many different TLAs, three-letter abbreviations. And so let's talk about the names that neutrophils have. Polymorphonuclear leukocyte. That means many shapes of the nucleus white blood cell. And if we look at the peripheral smear, we can see that the nucleus has lots of many shapes and sizes. We also notice those neutrophils have segmented nuclei. If you look at the nucleus of the cell, you can see it's divided up into little segments. And so that's why they call them segs, segmented neutrophils. And then the neutrophils belong to a family called the granulocytes, but there's lots and lots of neutrophils and not very many of the other granulocytes. And so you'll hear neutrophils and granulocytes used interchangeably because neutrophils make up the vast majority of the granulocytes. So when you see polymorphonuclear leukocytes, or PMNs, they're talking about neutrophils. And when you see the word segmented or SAGs, segmented neutrophils, they're talking about neutrophils. And when we talk about granulocytes, we're talking about neutrophils and eosinophils and basophils, but mostly neutrophils. So let's talk about the granulocytes. Neutrophils make up the majority of the granulocytes, a type of white blood cell. But there are other white blood cells that are granulocytes besides neutrophils, and those are the eosinophils and the basophils. When we do a right Giemsa stain on a peripheral smear, the stuff that takes up the red is the stuff that likes eosin, the red stuff, and so stuff that takes up the red stuff is considered to like eosin. That's where the word eosinophil comes from. And then basophils take up the other basic dye. The dye that turns stuff purple is basic, and that's where basophils comes from. And then if they take up equal numbers of both, we consider them to be neutral, and that's where the word neutrophils comes from. So I photoshopped this. You shouldn't see this many white blood cells under one peripheral smear, at least during the same lecture. But I put this in here so you can see this. So these are two neutrophils. If we look at the cell cytoplasm, they take up red, the eosin, and the blue, the basic dye, equally, and that's where they get their neutrophil word from. But this cell right here, this granulocyte, this white blood cell, takes up a lot of the eosinophil, the eosin stain, and that's where it gets the name eosinophil. So here are two neutrophils and an eosinophil. And these are all granulocytes. And here is a basophil. We don't see very many basophils on a peripheral smear. you'll see about 90 neutrophils before you see an eosinophil. You'll see about 99 neutrophils before you see a basophil. But eosinophils, neutrophils, and basophils, they make up the granulocytes. And if you look very carefully at a peripheral smear of granulocytes, you can see that they're full of granules. And those granules contain substances that are toxic to bacteria and viruses. That's how they get the word granulocyte, because of these little granules that we can see inside of the basophil, the neutrophils, and the eosinophils. I think I said this. The granulocytes contain granules. That makes it simple, and they carry all sorts of various kinds of poisons designed to kill infectious organisms. That's why people with neutropenia, pinia is just a word for too few, and so people who have neutropenia, they have too few neutrophils, they are going to be easily infected. And so if you work in a hospital and somebody has neutropenic precautions, that means they don't have enough neutrophils, and they probably don't have enough granulocytes, and because they have too few neutrophils, it's very easy for them to get infected with infectious agents. So somebody in the hospital on neutropenic precautions is something we'll talk about in the forum section. See if somebody is listening and can put neutropenic precautions somewhere in our forum section. So those are the granulocytes. There's other white blood cells that are not granulocytes. And those not granulocytes are the lymphocytes, which are very important, and the monocytes, which is where those phagocytes come from. And here is a lymphocyte right there. It looks like a plasma lymphocyte. It looks like it's been activated to make all sorts of antibodies. And where did my monocyte go? I'm missing my monocyte. Well, so what is blood made of? Stuff that carries nutrients, removes waste, supplies oxygen and carbon dioxide removal, stuff to make us stop bleeding, and then the star of our show, immunity. So please don't forget all the important things that blood does, especially this: combat infection and organize our immune system. So let's talk about hematopoiesis. Hematopoiesis is just simply how blood cells are made. And so you'll want to recognize that word and know that's just how blood cells are made. So when we talk about a peripheral smear, those are the cells that you should see in the circulating blood. The cells we're getting ready to talk about are only seen in the bone marrow on a bone marrow biopsy in a healthy person at least. I wanted you to be familiar with these two terms right here: the myeloid family, called the myeloid line, and the lymphoid line. When we talk about blood cells, they're either going to be of the myeloid line, and that's everything but the lymphocytes. The lymphocytes are in the lymphoid line. So if you can remember, the lymphoid line is the lymphocytes, and the myeloid line is everything else, then you will have a great understanding of these terms, because we will use terms like lymphocytic and myelocytic leukemia, and already you should be able to tell the difference between something that's lymphocytic and something that's myeloid or myelocytic. I want you to be familiar with the term differentiation. Differentiation is just quite simply to make different. As cells differentiate, they become different from the stem cells. Stem cells are how all cells originate, and then they differentiate. They change from that stem cell to become the final product. And so once the cell has become its final form, it is fully differentiated, meaning it has become completely different than its stem cell. The more differentiated cells are, the more they are like their final product. And we'll use differentiation and differentiated all the time when we talk about cancer and chemotherapy. All right, so all of the blood cells begin as a stem cell. They talk about stem cells all the time on TV and in the media. Stem cells are cells that you can make any other cell from. And the cells of the blood begin as a stem cell as well. These stem cells divide rapidly, making not only more stem cells, but cells that eventually differentiate into their final form. So when we talk about the development of any of the blood cells, they all start out as a stem cell. And the stem cell, when it divides up, can either make another stem cell or it can make something called the myeloid progenitor or the lymphoid progenitor. So our stem cell, and notice they look the same on the cartoon. We can do very special stains to see what the difference between these two are, but I wanted you to see the stem cell becomes two different types of stem cells. The myeloid line, which makes everything except the lymphocytes, which come from the lymphoid line. And I've shown you this slide. The myeloid line makes the red blood cells, the granulocytes, the monocytes, the platelets, and the lymphoid line makes lymphocytes. So if you can remember, lymphoid makes lymphocytes, myeloid makes everything else, you'll be well ahead of the game. Well, that's confusing enough. Here's the lymphoid line. Notice the stem cell at the top, and as we go down, this stem cell differentiates into its final form, the lymphocytes. And so let's take a look at this. So this is our stem cell, and the stem cell has become the lymphoid progenitor, and that will turn into a lymphoblast. Blast just means really immature cells. So when people are talking about blasts, they're usually talking about very immature looking cells. So our stem cell becomes a lymphoblast, and then pro-lymphocyte. Pro in the hematology business is the same thing as pre, and so we can consider this to be a pre-lymphocyte. So look for that blast word, look for that pro word. That pro just means pre, and blast means very immature. And notice its different appearance under the microscope. It can be very difficult to tell these cells apart unless you're trained to do so, and these are things that we usually only see under a microscope, at least these cells up here. So our stem cell becomes a lymphoblast, a very immature lymphocyte, which becomes a pre-lymphocyte, a pro-lymphocyte, and that can differentiate either into a B lymphocyte, which we'll talk about, a T lymphocyte, which we'll talk about, or a type of lymphocyte called a natural killer cell. There are lymphocytes in your body that are designed to seek out tissues that are ill and cancers and attack them and destroy them, and they're called natural killer cells. And then another type of B lymphocyte is a plasma cell. A B lymphocyte can be activated to make lots of antibodies, and when a B lymphocyte has been activated to make lots of antibodies, it's called a plasma cell. So I wanted you to be familiar with these terms because the literature throws these terms around like you guys know everything about this. Well, that was the lymphoid line. That was simple enough. The lymphoid line makes lymphocytes, and that's it right there. The myeloid line is very complicated because the myeloid line makes everything else. So our stem cell can differentiate into a megakaryoblast. A megakaryoblast will eventually become the platelets. Mega, big, karyoblast, meaning big cytoplasm. These are going to become great big cells that break apart into little platelets. The myeloblast is going to make monocytes and granulocytes, and then the pro-erythroblast, erythro, meaning that this is going to become an erythrocyte, a red blood cell. So the stem cell that becomes a myeloblast can make monocytes and the granulocytes. And we talked about basophils, neutrophils, and eosinophils. The myeloblast can become a monoblast, an immature monocyte, a pro-monocyte, which is a pre-monocyte, and then eventually a monocyte. And so this is the differentiation of the monocytes. The myeloid line can make the granulocytes, and I think we go through the neutrophils first because they're most common. Which brings us to this, granulocyte colony stimulating factor, filgrastim, Neupogen. It's abbreviated GCSF, granulocyte colony stimulating factor. This drug stimulates the myeloblast, stimulates the stem cell in the myeloblast to make more of these, make more granulocytes. And so somebody who's either getting cancer, chemotherapy, or for whatever reason is neutropenic, too few neutrophils, we can give them granulocyte colony stimulating factor to stimulate the production of neutrophils. Filgrastim, Neupogen, GCSF. Everybody in their nursing career, before they graduate from nursing school, will give somebody an injection of this, I assure you. This is a very innovative drug and very commonly used. Please do not confuse granulocyte colony stimulating factor with granulocyte monocyte colony stimulating factor. GCSF and GM-CSF are two different drugs, I assure you. People are confused about this all the time, and I hear stories about it, which is why this is in this lecture. So granulocyte, monocyte colony stimulating factor is sargramostim or leukine, GM-CSF. This isn't so commonly used as GCSF. I did want you to know the difference. All right, so when we stimulate the myeloid line to make more granulocytes, here's our stem cell differentiating into a myeloblast, and here promyelocyte, myelocyte, metamyelocyte, a band, that's where that banded nucleus, that's where that word comes from, and then it becomes neutrophils. When you talk about somebody who has an elevated white count and a left shift, they're talking about seeing these cells right here under the microscope on a peripheral smear. In a normal person, we should see neutrophils. But in times of infection, the body starts marginating, pushing out white blood cells quicker and more immature. And so when we see immature cells underneath the microscope, we call that a left shift, because in the old days when the hematologist used to count, we used to have little counters that we would count these on, and the bands and metamyelocytes and myelocytes, those counters used to be on the left, and that's where that word left shift came from. There's no such thing as a right shift, because we'd operate the microscope with our right hands and count the cells with our left hands. So when you talk about a left shift, they're talking about seeing bands and metamyelocytes and maybe even myelocytes under the microscope because of an infection. And I don't want to go through all this, but I did want you to see that the eosinophils basically have all the same words, other than they begin with eosinophil instead of neutrophil. And the same thing goes for the basophils, and they all look different as well. The myeloid line leads to platelets as well. The megakaryoblast, pro-megakaryocyte, becomes a megakaryocyte, and that breaks apart into little platelets. A megakaryocyte is actually a cell. We call the platelets cells, but they're really pieces of a broken up megakaryocyte. And then the myeloid line also makes red blood cells. So stem cell makes a pro-erythroblast, which differentiates into an erythroblast, normoblast, otherwise known as an orthochromatic erythroblast. And then something is important is a reticulocyte. Notice that the nucleus of a red blood cell disintegrates over time, and when it becomes an erythrocyte, it doesn't really need its nucleus anymore, and so it breaks it down. Reticulated means like a net like pattern. So a reticulocyte still has a little parts of its nucleus. This is a brand new, fresh red blood cell. So sometimes we'll get something called a reticulocyte count. That's a very special stain we can do to peripheral smear, and we can count the reticulocytes to see if the bone marrow is making normal amounts of erythrocytes. So if somebody's blood count is low, but we see normal amounts of reticulocytes, then we know that the bone marrow is making the red blood cells appropriately. Well, you want to know what erythropoietin is, erythropoietin, depending on who you argue with. Erythropoietin is synthesized by the kidneys, and it stimulates the production of red blood cells. Your kidneys are very sensitive to not getting enough blood supply, and when they sense not enough blood, not enough oxygen, they make erythropoietin. This is why people with renal failure, this is why people with kidney disease have anemia. They have low red blood cell counts, and quite simply, when the kidneys don't function, they don't function to stimulate the production of red blood cells. Well, we can take erythropoietin and make it a drug, a poietin. It's a drug that stimulates the production of red blood cells. A poietin is also Epigen, Procrit, EPO. I assure you, all of you going to nursing school will give somebody an injection of a poietin, Epigen, sometime in your career. Lots of lecture, and what have we talked about? 3 drugs. I think you'll notice this pattern as we go on. So I want you to know that everything starts as a stem cell, and the stem cell divides into either the myeloid line, which is all the cells except the lymphocytes, which come from the lymphoid line. And I think I've shown you this slide enough times. So let's talk about immunology. Now that you know all the casts and characters, we'll talk about the study of the immune system. All right, so every day the human body is exposed to numerous bacteria, viruses, and parasites. Our bodies are always fighting off these organisms. Our immune system is always fighting with bacteria and virus and funguses and parasites. Your bowels are full of all sorts of bacteria, and we need them there. They help digest. They're a major source of vitamin K, if not the most important source of vitamin K. So we use the bacteria in our gut as nutrients. And if we did not have an immune system, if we had immune deficiency, then we would succumb to infections very quickly. So our immune system is constantly at work battling and winning the battle over bacteria, viruses, funguses, and parasites all the time. All right? And our bodies are really good at fighting off an organism when they encounter it for the first time. But your body knows how to recognize organisms so it can launch an even bigger response next time that organism comes around. That's why many diseases they'll infect us once, and once our immune system recognizes those diseases, those bacteria and virus can't cause infection again because our immune system recognizes them. Not every bacteria and virus works that way, but many of them do. So our bodies are able to remember organisms so we can fight them even better for subsequent exposure. This is our strategy when we immunize people. So immunity is the body's ability to remember foreign substances and attack them even better the next time around. This is an important concept, the ability of the body to recognize these foreign substances and attack them even better upon subsequent exposure. That's our definition of immunity in here. Immunity is not the development of tolerance to a drug. People will say, oh, I'm immune to amoxil, it doesn't work anymore, try something else. That's not what that is. Immunity is what I told you it was, this definition right here. The body's ability to recognize foreign substance and attack them better the next time, attack viruses and bacteria even better because the immune system recognizes them. That's what immunity is. It's not tolerance to a drug. That drug doesn't work on me anymore. I'm immune to it. That is not how we use immunity in a professional setting. Or bacterial resistance to a drug. After time, certain drugs can be resistant to antibiotics. We don't say those bacteria are immune to those antibiotics. We say the bacteria is resistant to the antibiotics. So I wanted you to have a formal definition, a formal understanding of what the word immunity means. All right, so here's a cartoon I made a long time ago for a lecture, and the cartoon remains. Here is a virus, and I had the time to animate back then. So this is our virus floating free in the blood. And so the virus is going to enter a healthy cell and infect that cell. And notice our cell is not very happy about this. Notice that our virus takes its antigens, its outside, we can call those the viral surface antigens, and they will be expressed on the surface of the cell. This is part of how our body stimulates the immune system. When the virus enters the body, antigens are expressed on the surface of the infected cell. And then the viral DNA and RNA, the viral genetic material, goes inside of the cell to reproduce. So let's see if we can see that on instant replay. Sick cell. Oh, going backwards. Virus enters the cell. Surface antigens are expressed at the surface. Genetic material goes into the nucleus of the cell to reproduce. Bacteria have everything they need, all the machinery they need to reproduce on their own. But viruses rely on the body's genetic systems, its biochemistry, to reproduce. So the virus uses the body to reproduce its parts. Bacteria have everything they need to reproduce. They just need a warm, wet environment with lots of sugar. And so now the virus genetic material has gone into the nucleus of the cell and reproduced, used the cell's genetic engineering to reproduce the genes of the virus. Fortunately, we have several types of immunity to protect us from these germs, that virus. They talk about cellular immunity, which usually involves cells. And then they talk about humoral immunity. Humoral just means the liquid part of blood that comes from way back in the old days. But when they're talking about humoral immunity, they're usually talking about antibodies floating in the bloodstream. When they talk about cellular immunity, they're talking about cells of the immune system. So let's see if we can make this simple. Let's talk about cellular immunity. All right, so remember that's my virus floating freely in the plasma, the serum of the blood. And a macrophage comes and eats the virus. Maybe the macrophage senses the complement and some antibodies attached to the virus. And so the macrophage comes and consumes the virus. And there's a nice electron microscope of a macrophage. We can see its little feet sticking out and feeling. That's a red blood cell back there. We'll see the little macrophages coming and finding the bacteria. So when the macrophage expresses viral antigen, when the macrophage consumes the virus, it too expresses the viral antigens on its surface. and when the macrophage does that, it expresses the viral antigen on the surface with something called a major histocompatibility complex. Major means it's very important. Histo means tissue. Compatibility means how all those cells get along. And complex means it's not this simple. Actually, it's a huge complex, like a city of different types of proteins that make up the major histocompatibility complex. The major histocompatibility complex is basically how immune system cells are able to identify themselves from foreign tissue. And it's a way that the cells can signal each other, the major histocompatibility complex. So again, my macrophage consumed the virus, expressed viral antigen on its surface with major histocompatibility complex. And when that happens, the macrophage secretes something called interleukin-1. All right, so the macrophage consumed the virus, expressed viral antigen on its surface along with major histocompatibility complex, and secretes interleukin-1. Interleukin is short for interleukocytokine. Inter between leuko, white, cytocell. Kine means movement, how it moves, how they communicate and tell each other to move in this direction. So interleukocytokines are how white cells communicate with each other. Now we know that other cells of the body can make interleukins too. But for those of you reading a majority of the stuff out there, interleukocytokines are the same thing as interleukins, and this is how the immune system is able to communicate with other white blood cells and with the body. So interleukin-1 is how the macrophage calls for help. Interleukin-1 attracts other macrophages. But interleukin-1 also attracts T helper cells. T helper cells are also called CD4 cells, cluster differentiation 4 cells. T helper cells and CD4 cells are the same thing. You should know that now, especially before we talk about HIV and AIDS. T helper cells, CD4 cells, the same thing. They are a type of lymphocyte, and they are the major mediator of cellular immunity. So when our macrophage has expressed antigen with the major histocompatibility complex, and it calls for help using interleukin-1. And what senses that? It's T helper cell. Here I come to save the day. T helper cell is on his way. And so T helper cell is responding to the call of interleukin-1 to help fight with these viruses that have attacked the body. So T helper cell is the same thing as TH, same thing as CD4. You'll see all these abbreviations, especially when we talk about HIV and AIDS. And T helper cells are drawn into the area. They're attracted to interleukin-1. And so here's my T helper cell, and it has been attracted to the area because the macrophage has secreted interleukin-1, which attracts T helper cell to the area. And T helper cell is able to recognize the viral antigen and the major histocompatibility complex. And when the T helper cell recognizes that, it will release not only interleukin-1 but interleukin-2, which flew off our screen. And interleukin-2 attracts even more T helper cells and pulls them into the area. And T helper cells are very essential in fighting these bacteria and viruses. So let's review. The macrophage ate some kind of microbial product, whether it was a virus or a fungus or a bacteria. It presented antigen. You'll hear about antigen presentation with a major histocompatibility complex, and it will secrete interleukin-1, and this will attract T cells and activate T helper cells, and they will come to the area, and when they identify that antigen presentation, they will secrete interleukin-2, and now everything's completely stimulated. All right, so this should all be in yellow. interleukins, there are many interleukins. If you get on Wikipedia, which is fine for information on interleukins, they list 30-something. I was going to talk about some of them, but there's no point, so we'll do that in the forum section. There are many interleukins, and it's important that you know that interleukins are how the white cells in the tissues communicate with each other. But interleukins are also used as drugs for all sorts of reasons. So for those of you behind in the forum section, immunology is a huge field, and we could put hundreds and hundreds of postings in the immunology section and not even scratch the surface, but I know everybody's tired at this point, and my interleukin section doesn't get very full. So let's get into the forum section and see how many interleukins we can use as drugs to treat things like cancer and multiple sclerosis and things like that. Speaking of that, interleukins, a type of interleukin are interferons. Interferons just quite simply interfere with a virus's ability to replicate its DNA and RNA. So we'll talk about interleukins and interferons in the immune part of our forums. Interleukins and interferons are naturally occurring substances that our bodies use to fight infection, and we can reproduce those substances and use them as drugs. All right, so cellular immunity is basically based on T helper cell being in the center of amplifying the immune system. Cellular immunity and humoral immunity are not separate things. They are actually part of immunity, but we divide the parts that rely on cells, we call that cellular immunity. Parts that rely on antibodies recognizing things in the bloodstream, we call that humoral immunity. So when we talk about humoral immunity, we're basically talking about antibodies recognizing things in the bloodstream. Here is a B lymphocyte. It'll be coated with these antibodies waiting. They're like receptors waiting for a specific type of antigen to activate them. And so here is viral antigen. It can be either loose or attached to the virus. It doesn't matter. And here the B lymphocyte is able to recognize the specific antigen that it recognizes. And when it does, it can either recognize that antigen free-floating in the blood, or it can recognize the antigen being presented by the macrophage. A neat little trick the T helper cell can do, it can present antigen with major histocompatibility complex as well. This is one of the way T helper cells help, is to present antigen as well. And so when the B lymphocyte senses antigen, either presented by the T helper cell, the macrophage, or antigen floating freely in blood, when the B lymphocyte recognizes any of these things, what will it do? It will become a plasma cell and make even more and more antibodies floating in the blood, so that we even have an increased response against viruses and bacteria. Although this B cell is going to be very specific for this type of antigen. So these antibodies are going to stick to the specific antigen and mark it, or opsonize. That's what we call it when something is coated with antigen. It's been opsonized, and this makes it easier to be attacked by granulocytes and monocytes. So coating something in antibody is a great way to stimulate the immune system to come get rid of it. When we talk about antibodies, they'll talk about light chains. We've divided the light, my little Y-shaped protein, into 4 little parts. 2 sets of light chains. We call those lambda and kappa. Kappa and lambda chains are the light chains. And then there's also heavy chains of the antibody, and we can talk about that in forum section. The top of the Y is where antigen binding occurs, and the bottom of the Y is what binds to the immune system. This part signals the immune system to work. This is the part that binds to antigen. And I think I said this. Please do not confuse antigen, what stimulates the immune system, with antibody, the little white-shaped proteins that I'm talking about. Well, so there's my virus again, and the first time the macrophage noticed it, it attacked it, but next time that virus decided to come around, there were all these antibodies waiting to attack it and coat it. And this is how immunity works, and this is how immunizations work. Immunizations work by making sure that anytime this type of virus ever comes to attack us, we're full of lots of antibodies to attack that virus, so the macrophage in the immune system can immediately consume it and get rid of it without it causing infection. And so we've made the full circle. The first time the macrophage ate the virus was coated or marked by complement and a random assortment of some antibodies. And this was the initial reaction to foreign substances. But the second time the macrophage ate our virus, the virus was coated or marked by a large number of these antibodies specific for that antigen. The secondary reaction to foreign substances is much quicker and much greater, and what defines our immune system. And so that's our definition of immunity. The next time this virus comes to attack us, our reaction is much quicker and stronger because the body's immune system remembers those antigens and has made all sorts of antibodies and B cells and T helper cells able to recognize those antigens and react even quicker the next time around. So here, our log of antibody titers, how many antibodies we have is on this axis, and this is over time. And the first time we're exposed to something, the first thing that goes up are a type of immunoglobulin, a type of antibody called IgM. We'll talk about that, our acute antibodies. And then notice our IgG goes up a little bit and then goes back down. But the second time we're exposed to an antigen, sure, our IgMs go up and down like they usually do, but our IgGs go up and stay. And so we're getting ready to talk about what this means, making immunoglobulin G even more available for second exposure is what we've been talking about. So let's talk more about those antibodies. We talked about cellular immunity when we talked about macrophage ingestion, granulocyte toxicity, T helper cell help. And then humoral immunity had to do with antibody production, very simplified version of this. So antibodies are made by B lymphocytes to mark antigens for phagocytosis. There are several types of antibodies. IgM was going to look like 5 immunoglobulins. We abbreviate immunoglobulin Ig. They're just antibodies, abbreviated AB. And so IgM is a type of antibody, and this is our initial or acute reaction to infection. It's called a primary antibody. So when somebody talks about getting acute antibodies for a certain disease, they're talking about measuring IgM levels because when we're exposed to a disease, the first thing that the body does is generate these IgMs. IgG looks like a little Y-shaped single immunoglobulin. We call that the chronic Ig. When you're immune to something, if they want to see if you're immune to something, they'll check your IgG levels because once we're exposed to something, our IgG levels go up and stay. And they're considered the secondary or the memory antibody. So know the difference between IgM and IgG. Those are the two most important antibodies for you to know the difference. Antibodies, immunoglobulin is the same word. All right. We can also use antibodies as drugs. Hepatitis, breast cancer, the list is long. And there's all sorts of diseases that can be treated with IgG antibodies. And when you go through, I want you to remember that some antibodies can be used to signal the immune system. Some antibodies will attack and stick to foreign substances. and signal the immune system, but we can also use antibodies quite simply as drugs to activate and deactivate receptors. If you remember Graves' disease, we make antibodies by mistake that our body thinks is thyroid stimulating hormone. These antibodies are not attacking the thyroid. The antibodies are just signaling the thyroid to produce more thyroid hormone. So when you go through and talk about antibodies as drugs, think about, is that antibody being used as part of the immune system, or is that antibody just quite simply being used to activate receptors? And we can talk about that. We could have 1,000 postings about this in the forum section, and hopefully I'll get at least 10 or something. There's IgD antibodies. Those are singular immunoglobulins that are attached to B lymphocytes. If I had the time, I would just coat this B lymphocyte with all these little IgD antibodies. These antibodies attached to the lymphocyte are what the B lymphocyte senses antigen so that the B lymphocyte can make more IgGs. You'll see IgA antibodies talked about. They look like two little immunoglobulins tail to tail. These are what are in secretion: tears, saliva, intestinal fluids. IgA is what protects our mucus secretions, protects our orifices from infection. As well as IgE is an antibody, and this is going to attach to mast cells. Mast cells are what cause histamine release. We talked about histamine release in the histamine, serotonin, and ergot section. And so when we have hay fever, when we're allergic to things, these little mast cells that are stuck to these little mast cells have IgE antibodies, and when things that we're allergic to come in contact, things we're allergic to, those antigens come in contact with the IgEs, histamine is released from the body, and we have allergic reaction anywhere from hay fever to anaphylaxis. So when the IgE on the mast cell comes in contact with antigens or allergens, those are the same things, the mast cell releases histamine, which increases capillary permeability. It attracts white cells, and this is the initial cause of hay fever, which is sneezing and runny nose and itchy eyes, or severe bodily allergic reaction, a systemic allergic reaction, which is anaphylaxis. All right, so we talked about T lymphocytes. A type of T lymphocyte are the CD4 helper cells, the T helper cells. There's also the natural killer cells. We talked about those as the cytotoxic lymphocytes. They're also called CD8 cells. These kill viral infected cells and tumors by stimulating something called apoptosis. Early on, when our virus attacked our cell and made the frowny face on our nucleus, unfortunately that cell could not be saved. What happens is a cytotoxic, a CD8 T killer cell will come and see that that cell is infected, and it will stimulate apoptosis. Apoptosis is like the self-destruct button on a cell. All cells have the ability to self-destruct, and that's what apoptosis is. And so a T killer cell will go up to a cell and cause it to have apoptosis, causing it to self-destruct. Suppressor cells shut down that T cell mediated immunity. When we talked about cellular immunity, everything was ramping up, cascading in the upwards direction. And suppressor cells, once we've had enough immune response to something and our immune system has succeeded in averting that infection, fighting off the infection, suppressor cells come through and shut down T cells, shut down the T helper cells, shut down cellular immunity when that reaction is no longer needed. And they too do it through apoptosis. They'll come through and tell cells to self-destruct. That's a normal part of bodily function. Then there's the B lymphocytes. The B lymphocytes will recognize antigen and either divide and or make antibodies. And so the plasma cells, when a B lymphocyte is stimulated, it can become a plasma cell and just make lots and lots of antibodies, or it can reproduce many times and become memory cells, which are on guard for future exposure. And when those are exposed, they can become plasma cells and make lots and lots of antibodies. Sure, immunology is a lot more complicated than this, but this is the best I could do in 1 hour. And that's certainly more than enough for one day. Be sure and check out what's going on in the forum section. That is a great place for those of you who are behind to get caught up, do a bunch of extra postings, get a great handle on all the things that we can do with the immune system and the drugs related to the immune system. When I come back, we're going to talk about HIV and AIDS. Until then, aloha.
This is Cable 55, UH Maui College Television Network.